back to the mechanisms for adding genetic information. You were asking about the mechanisms for additional genetic information, and I just have to see if this will follow. Yeah. Just have to point out the right place. Here we are. Creationists often claim that no known mechanism can add new genetic information, but if you look at the scientific literature, there are actually several known mechanisms that can add new genetic information to an existing genome, and I'm going through a few of them. Within the last 2 decades or so, it has appeared to scientists that actually there is brand new genes called de novo genes, or orphan genes, which appears from non-coding DNA. So you have a protein coding gene where no protein coding gene was in the ancestors of this.
Okay, hold it. Let's take it. I want to take these one at a time so nobody gets confused. Are you saying there were new genes that came from non-coding DNA? Was this new information added to the DNA, or silent genes that were activated that were already present? Are you saying there was new information added, or just something that was normally non-coding all of a sudden started doing its job? Someone came off of welfare, in other words.
I think I have to explain how these were identified. I think that would answer your question, if I may.
Okay.
Go through that. Just to make sure that that our audience are familiar with what a gene and what a protein coding gene is and how it works. DNA is built of nucleotides. It's this well-known double helix structure, and all the little balls in the middle there are representing the so-called nucleotides. There are 4 different nucleotides called, normally abbreviated A, T, C, and G.
Right.
And the function of a protein coding gene is that the DNA is transcribed into a so-called messenger RNA molecule, and that molecule is translated into a protein. It's a complicated mechanism, of course, but that's a short story of it. And it is the protein that has the function. The gene itself, the DNA gene, doesn't have any other function than being transcribed and translated into the protein, and it's a protein that actually has a function. This is going on by taking 3 nucleotides at the time and translating it into 1 amino acid, and a sequence of amino acids then builds the protein. So it is the sequence of amino acids that decides decides what the protein will be doing. We'll skip this one. Then identification of candidate new genes. This is, I think, what you were asking. How do we identify a new gene?
No, I'm asking, not is it a new gene? Is there new information added, or is it a scrambling of existing information, or a gene that came off of welfare and decided to get to work now?
Yeah,
it's already there.
This is more or less the same. What people have been doing for identification of these new genes, they have compared a number of very closely related species, and often it's species within the same genus. Often they have been using Drosophila, coming right up there. Drosophila is a fruit fly,
right?
And there are a large number of, a very, very large number of Drosophila species, and I think that we can agree that they had a common ancestor, which was a Drosophila fly.
Fly, yes.
Just to make sure that we know what we agree on. And then a number of these different Drosophila species have been analyzed in very, very great detail. The most well-known one is Melanogaster. That's the one that has been used for genetic research for decades, or almost 100 years now. And in melanogaster and its closest relatives, or the species closest related to it, researchers has found a number of protein coding genes that are not present in any other Drosophila species. And there's a very, very big number of Drosophila species that has been investigated here. So the simplest explanation is that these genes actually occurred in the common ancestor of melanogaster and its very, very close related species, and that it is not a gene that has been lost in any of the other species. It has never been there. It has been the sequence that make up this gene is present in the other species, but it is not an active gene, and it has never been an active gene. It's not a sleeping gene that just waits to be activated. It's a sequence that is not that doesn't have the right signals for being translated into a protein. Actually, what they did in this particular investigation here was that they were able to identify the so-called stop codon. If I go back a few slides here, a protein coding DNA sequence is defined by its start codon, which always is ATG, the start codon, and then a number of amino acid defining codons, and then a stop codon. And between those two is the protein, to tell the story very short. And what they were able to do in this investigation, in this slide, come on, here, and no, this one, this one, and this one. Those two are both Drosophila investigations. And what they were able to do is to identify the mutation that removed a stop codon in the middle of this DNA sequence, so that instead of a stop codon, you have an amino acid defining codon, and then what was a non-coding DNA sequence suddenly was turned into a coding DNA sequence, and now the protein was able to be translated and function. So if that is not new genetic information, I don't know what you are asking for.
Okay.
This is a protein that is produced in some species that has never been produced anywhere before. They have tried to search the entire gene bank, which comprises millions and millions of DNA sequences from hundreds of thousands of different, perhaps not hundreds of thousands, but tens of thousands of different species. And they haven't found one single example of this particular protein in anywhere. So it is a new protein.
Well, it's a new protein, possibly. Let's discuss this for the listeners here. What the stop codons would be similar to a period when you're writing the English language, and I'm assuming it's the same in Danish or Dutch or German or French. You have a sentence, a single thought, and you put a period at the end, and now you start a new thought. If by accident one of the periods was taken out, you would put two sentences together and get in English what we call a run-on sentence, which now may give a little slightly scrambled information to the person trying to read it, like what does this mean? So I think what you've done is you've discovered that in a certain few cases with the Drosophila fly, the stop codon, the periods were removed, and it developed a run-on sentence. And they're calling that a new protein. Okay, let's call that a new.
They're not calling that a new protein. It is a new protein.
It is. It's a new protein.
You have to understand this. Before this stop codon was removed, there was no protein. No protein was produced.
Right.
Only after the stop codon was removed, a protein was produced.
I would like to point out that this is not new information. The information was already there. It is now scrambled into a run-on sentence. And I'd like to point out, it is still a fly. In every case, it's I mean, are you telling me there's been examples where it turned into something that was non-fly?
May I remind you, Mr. Hovind, that at the very beginning of this, I said that, yes.
Have to wait longer. More time.
In observable time frame, we will not expect to see a dog produce a non-dog. That was what I said. And you insist that that is what we should expect to see. Actually, you insist that in order for you to accept evolution, we should observe what the theory of evolution says that we should not observe. That is nonsense.
Okay, I am pointing out for the listening audience here that what you've given us is an example of removing a stop codon, creating a new protein, which I will give the analogy of a run-on sentence, which would be a whole new thought. Two sentences run together, like, whoa, it's a new sentence. I agree. It's not new information.
The information before that new sentence was not two sentences. It was 2 times nonsense.
Right. So that is not a mechanism that's going to change a single-celled organism to a human. This is not observed, and you fall back on your God, which is time. Time can somehow solve it. This was a loss of information. Have any of the flies that have been observed to have this new protein, first of all, have any of them been an improvement over the fly? Secondly, who are they going to marry? Or is this new protein going to be lost in the next generation? Because you'd have to have 2, a male and a female, in the same place at the same time and be interested in each other. You've got a host of problems. If this happens once by random chance in one fly gene code, how's this going to get passed on? Has it ever been demonstrated that it gets passed on? And is it new? Is it helpful to the organism or harmful? They've got flies that are blind, flies that can't fly, flies with curled wings. They don't get an improved fly.
Well, actually,
you believe it.
Some of these identified new genes actually has been proven to be essential to the fly. Probably because it has lost some other gene and this new gene has replaced it, or because some other function has developed or had evolved in the fly so that it became dependent on these genes. So actually some of these genes are now essential to the species where they are found. So yes, it is actually new information, and the fly has changed the new species. It's not a non-fly, which you should not expect it to be. It's a new species, which is what you should expect it to be. And just to make a little, well, what do we see here? There's a number of pictures. What do we see?
I see a variety of dogs, but let me point out.
No, you don't. Mr. Hovind, no, you don't.
Oh, okay, there's a panda bear in there. There's a panda bear.
No, it's not. No, it's not a panda. That's a dog. But the one to the left is a cat.
Oh, okay. But Stefan,
apparently, Mr. Hovind, apparently cats and dogs has an overlapping variation.
Whoa, they both have 2 eyes, and they're both affected by gravity. That proves it. Stefan, hold it. Yeah. We started late because of the technical problems, and I apologize. I'll do this again anytime. I have another debate, so we have to stop in about 15 minutes, and then I have to stop. I have another debate already scheduled. So yes, good. You got a cat with long hair that I mistook. I just glanced at him, mistook it for a dog. That's, I think, evidence that the same designer made them. Some people confuse the Porsche and a Mercedes. You know, when they go by at 200 miles an hour. Well, what was that? Well, they both have 4 tires. They got to be related. No, it's the same. It's a common design, and I don't think I'll ever get you to see that. But I want to go back to your fly illustration. You're getting a new protein from this stop codon being removed. It is still a fly. You still have a problem of this being passed on to the next generation. And where is the example of an improvement of changing to anything other than fly? This isn't help for your theory.
We shouldn't see anything other than a fly within historical time.
So because we don't see evolution happening, that's proof that it happened.
No, we see evolution happening. We see new genetic, new proteins, new protein coding genes appearing at these flies, and we have seen them all over the place. It has been. This is another example of Drosophila. This is humans. This is mouse and rat, and this is fungi and mollusks and tardigrades and nematodes and plants. It has been found all over the place. And Mr. Hovind, this is actually the mechanism that you were asking for. Because if within a time frame that is short enough for an organism still being in the same genus, like the Drosophila here. Drosophila is what we normally call fruit flies.
Right.
Within Drosophila, we have several examples of new genes. 195 new genes has been identified in various Drosophila species. Just think for a second. If 195 new genes can emerge within a single genus, then how many thousands can emerge within a family or within an order? And that is what you are asking for. New genes that change the genome of the organism. And we see it right in front of us.
Hold it. You're moving the goalpost here. Let me ask you this. I'm just maybe totally unable to give the information. How many genes are there in the fly's gene code?
About 20,000.
20,000. So
yes,
you're
so
saying
this is actually 1%. This is actually about 1% of it.
So out of 20,000, you're saying 1% of the genes have been modified through losing a stop codon. Again, they're losing information. They're not gaining information.
That's nonsense. Losing a stop codon is not losing information. It's changing one nucleotide into another nucleotide so that a stop codon turns into an amino acid codon. It's not losing information.
And are you convinced that that mechanism you just described of losing a stop codon and creating a new protein, that is the mechanism that is going to, over trillions of years, going to change a single-celled organism to a human and an elephant and a whale? Is that what you believe? This is the mechanism that will do it?
Plus a number of other mechanisms.
What other mechanism?
Well, we don't have the time for it, but I have a number of slides showing other mechanisms too.
Go ahead.
There are plenty to take up, but if you think about it, 1% within one group of organisms that are so closely related that even you would accept that they had a common ancestor.
Right.
So if you just for a short second accept that this could be extrapolated to family order, then you would not have 1%, you would have 10%, 50%. You would have almost all those genes that are not essential for the very basic biochemistry of the animals. Okay.
Let me give an example. When they first started the Olympics, I think 1930s or something, Hitler revived the Olympics idea. We could look at the top speed of people running, and let's say someone could run the 100-yard dash in 10.5 seconds. Over the last 70 years, they've got it from 10.5 down to under 9 seconds now, I believe. If in 70 years we can knock off 1.5 seconds, does that mean if we go 700 years, they can knock off 10 seconds and go down to 0 and run the 100-yard dash in 0 seconds? Is this logical, or is this stop codon, is this new, this protein mechanism that you just described, could it be that that has a limit? And you got 195, maybe they're only going to get up to 250 and something's going to shut it off, or the fly is going to die. You haven't demonstrated, other than imagination, SpongeBob, that maybe if we give it more time, there's your God, it could do it. This isn't science to say that. You can't go back in time. We can't go forward in time. All we've observed in all of human history is flies produce flies. If you want to say they have new information, did the fly get something different than a wing, or did it get a new structure? It had a new protein. What good did this do for the fly? Can it fly faster? Can it produce more offspring? What beneficial mutation has come from these 195 new genes in the fly out of the 20,000? Have you demonstrated any new information that's helpful to the fly?
Well, as I said, a few of them have actually turned out to be essential for the survival of the fly. I don't know if they have been analyzed so that people know what they actually do, but they have been demonstrated to be essential for the survival of the fly. So in some way, they are very, very important, some of these.
Well, now hold it. Are they essential to the survival of the fly because it is now encountering some new environment, like some man-made toxin or poison, or something in the atmosphere or the food? The world is constantly changing, sometimes man-made, sometimes nature-made. But did the fly ever get a turbocharger? Did he ever get a jet engine? Or is it still a fly?
As we should expect under the theory of evolution, it is still a fly, yes.
Hold it. As we should expect under the theory of creation, it is still a fly. Creation predicts that also, by the way, Stefan. Creation predicts animals will always bring forth after their kind, and a fly is the same kind of animal as a fly. So this is predicted by the creation model, not by evolution.
I would love to discuss phylogenetics with you and show you why phylogenetics is not, as you describe it, just lines on a piece of paper, but that it is actually very, very well founded in observation, because that would be what you are asking for. How do we know that different animals are actually related? What are the evidence that organisms or animals that you would say were different kinds, even though you have never been able to demonstrate that you actually know what a kind is. On the contrary, you have been contradicting yourself all the time on that, showing that well, I have to be blunt with you. You have been showing that you have no idea what you are talking about when you mention the word kind.
Okay,
but,
and
back to the question of this.
You have no idea what you're talking about when you mention the word evolution. You have no idea what you're mentioning about talking about when you talk about animals producing new information. That was not new information. It already had the sequence of letters. It removed a stop codon. It happened to make a new protein, but the information was already there. You didn't add anything to the gene code. You have no clue what you're talking about. Now, we only have 6 minutes left. Can you give a summary of your position?